Today, we're very happy to have with us Linda Tempelman, PhD. She's the CEO of Persista Bio, and she's presenting a continuously oxygenated macro encapsulation system enabling high-density packing and delivery of insulin-secreted cells. And some of her associates are listed as well on the slide: Tung Pham, Phuong Tran, Simon Stone, Christopher Picariello, Alan Lee, James Flanders, and Mingling Ma, who we've had on the Sugar Science prior. So it's a very interesting talk ahead, and a little bit short bio for Linda. She is leading Persista Bio into large animal proof of concept and product development. Persista Bio O2 line is an oxygenated cell therapy system that sustains compact implants that free patients from immunosuppression, and the initial indication is type 1 diabetes. I will drop a recent paper and a couple of related papers in the chat, and she will take questions at the very end. Thank you, Linda, for joining us, and we're excited to hear what you have to share.
Thank you very much. I was wondering if people would mind coming on camera for a minute and just waving and saying whether they're in the cell therapy field. So come on camera and wave.
Okay.
Hi, Shanine. How are you?
How you doing?
Good. You already heard this talk at the breakthrough meeting.
Yeah, but if each time you say something, I learn more.
Excellent. And then I'm just curious, who else is in the cell therapy field directly? Wave your hands.
Well, we've got Mark Fisher-Colbury, Ahmad Rita, Alice Smagorinsky, Charles Putnam, Paran Shokori, and Scott Ajax. So this talk will also be seen by quite a large audience once we post it to our YouTube channel.
Thank you very much, everybody. If you're off camera, I assume you're eating your lunch, and I empathize with you because my lunch is delayed. So I will be eating lunch after this. So today I'm presenting the results from a preprint paper that the link is available for. This work was done in collaboration with Th1nk Incorporated and Cornell University. I'm one of the authors, but the authors that did the rat experimental work are listed, and I've predominantly worked on the oxygenator part of it. So as Monica introduced, we're working on an antifibrotic oxygenated capsule for cell delivery, and our first indication and our preliminary data is in using pancreatic islets. Okay, so basically we're trying to do a proof of concept using allogeneic islets on a new type of cell therapy. And our goal here was to conduct rat proof of concept studies to show viability and function with a small form factor device with in situ oxygenation. And we're trying to do things a little bit differently. We're not trying to do the minimally viable dose of islets. We're trying to show that when you make the islets compact in a device, that it would scale up to large animal and human size. As I said, this is a collaboration between Th1nk and Th1nk Labs, where I worked for many years, an electrochemistry company, the Cornell University lab of Mingling Ma, and now Persista Bio. Quick, of course, for disclosures, a number of us hold stock in Persista Bio, and a number of us are on the patents that Persista Bio is licensing. And Dr. Ma is on leave from both Cornell and Persista Bio to work in industry. So I don't know everybody's background, so I'll give the very brief background. As all of you know, there are advanced therapies for type 1 diabetics, the closed loop insulin pumps, the artificial pancreas, the glucose sensors. But in this 2019 review article by Esther Latrace et al., they're still showing that you're not getting perfect glucose control with those systems. I'd be interested if people have newer references where some of the newer systems are getting better at glucose control. I'm not following that closely myself. But we do know that many, many people have shown very, very good glucose control with cadaveric islet transplantation. As all of you know, there's been difficulties in the US. There's one company in the US and one site, the CellTrans company, that's able to do that because they got a BLA. In other countries, islet transplants are more prevalent. Of course, islet transplants means there's less tissue available because every patient needs 2 or 3 cadaveric pancreata to get a better improvement. And of course, the patients have to take immunosuppression. I estimate 95% of type 1 diabetics can't have immunosuppression. It's just not called for. Their diabetes is not severe enough. So we're looking to do a new system, and we're looking to implant the cells. We're doing our proof of concept with pancreatic islets, but of course, the goal is to move on to stem cells. So we'd like robust glucose control, no immunosuppression, And, there's been a few newspaper articles and testimonials from patients that have had either islet transplants, or there was a testimonial in the Washington Post from a patient that was in the Vertex cell trial, just saying how different your life is if you have an implant and you stop measuring glucose and shooting insulin. And then just a super, super brief summary of what's going on in the field. I believe, this is my hypothesis, that the cells that people are developing are starting to work. So there's a number of companies that are developing those, as you see on the right. I'm sure some of you are very familiar with those. Other university efforts to make newer stem cell lines that may be even better. So I estimate that companies are spending hundreds of millions of dollars developing the stem cell line, and I don't think they're putting very much effort into the implant systems to protect the cells. There's been several implant trials that have not met their endpoints. So ViaCyte, before they were acquired, did several trials with encapsulation with holes punched in the capsules. Vertex has just closed an implant trial, and Cernova has not had the best successes with their system. So we believe that there's better things that are needed. I think there's gene editing approaches that may come along, and we're all excited to hear what Sana Biotechnology presents at the upcoming ADA meeting. They gave a little teaser in January that they'd implanted one patient with a genetically engineered human cadaveric islet. So this is kind of where we're at right now, and we're trying to go down the route of encapsulation with oxygenation. So I'm going to make the claim that I think that effective macroencapsulation may be the fastest path right now to take the current generation stem cells to patients. So that's what we're all here for. We're all here to take things to the patient. I think there's other approaches. I think there's going to be second and third generations. I think there may be more than one brand that comes out. But right now, we think if you encapsulate the cells, protect them from the immune system allogeneically, we have evidence that an oxygenated implant of our type has good chances of success. As I said before, this is a collaboration between Cornell and Giener. And basically during the pandemic, Mingling Ma called us up and said, hey, my stuff is working pretty good. My cell encapsulation is working well, but I have not the best results I've gotten have been when I've added a little bit of oxygen chemically, temporarily, and our small animals work better than our large animals. Let's collaborate. So historically, this is back into history for you, and you guys can look up the papers. The Mingling Ma lab at Cornell worked on a string system. They worked on nanofibers. Giener Incorporated back in 1999 was working with Clark Colton's lab and Gordon Weir's lab and doing an oxygenation device for some in vitro work. Giener worked with Therasite style capsules back in 2016, 17, and 18. And now we've come together and we're collaborating. Preprint published this paper, and we've formed a company to take this to market. Basically, right now, I'm calling it Gen 3. So basically, we've combined these with wearable electronics for proof of concept. And then we'd like to move to a fully implantable system for large animal testing and GMP manufactured for clinical use. So go ahead and look up the references if it interests you, but this was the string device that the Cornell lab developed. Basically, they had a string down the center and the islets were in a hydrogel on the outside. So the string was meant to give some rigidity and form to the string so you could pull the encapsulation in and out. They had a number of papers with different collaborators. Then they worked on a core shell nanofibrous device. So the cells are actually encapsulated because there's a nanofibrous capsule on the outside shown in blue or purple there, and that holds the cells in. So there's no chance of the cells sloughing off from the hydrogel. So they worked on the nanofibers, they worked on a switterionic coating that's patented, and they've published on the combination of those twos. So Gen 3, where our collaboration takes off, we took the core shell nanofibrous device shown in the center panel in beige. We combined it with the Giener oxygen generator, which is abbreviated IEOG for implantable electrochemical oxygen generator. You'll see that the nanofibrous device from Cornell is quite a bit different than what some other people in the field have used. It's not at all like the Therasite and Therasite-like EPTFE capsules. It's quite different. We think it has advantages because it doesn't have edges and seams and so on. And in one of these presentations, I don't think it's the one today, if you take the capsule, you can actually wiggle it like a piece of spaghetti. It doesn't kink up, and we've got some special features in there to prevent it from kinking. So the current system, Gen 3, the rats are wearing a wearable backpack. We're usually using large 300-gram animals so that they can carry this backpack around. And this has power for like 5 days, and then the backpack battery gets changed out. So a little bit about the oxygen generator, where I'm an inventor. So here's the latest batch of oxygen generators we've fabricated at Th1nk Incorporated. So basically, it's a patented device. Several components of it are patented. It's a proton exchange membrane type electrolyzer. So an electrolyzer takes water and splits it into hydrogen and oxygen. And there's no liquid acid inside this device if it were to leak, because the proton exchange membrane has protons on it that act as the acid, but it's just a water-based system with protons. We harvest water through the white membrane in the bottom picture. Basically, to electrolyze water, you need power, electricity, and you need water. So we've been harvesting water from the body. Only vapor phase water goes through that membrane. It's not Gore-Tex, but it's like a Gore-Tex membrane. It only lets vapor through a vapor transport membrane. And for right now, we have these external electronics that plug into a device. And we are already trying to move towards things that will be clinically acceptable. So it's already packaged in an ultrasonically welded titanium case, like other defibrillator and cardiac and pacemaker type devices. So here I'm showing a little bit of implant with the oxygen generator. So on the bottom left, you see the oxygen generator in situ. The device is about the size of a dime, maybe 2 dime thicknesses. We've not had adverse tissue reactions. And these devices have been running in rats for up to 90 days, as shown in the graph on the right. On the benchtop, we've had units running over a year with no difficulties. This core technology is basically the same technology that Th1nk Incorporated uses for nuclear submarines. So this technology has been around for 30 years in its present form, and Th1nk holds patents, but also proprietary secrets on the best way to make these catalytic electrodes. So we've had the stable production, as I mentioned, and this device, by changing the current, it's a current controlled device, and the amount of oxygen is proportional to the current. So if you want to use this in a rat study with a low amount of oxygen, you turn down the current. If you're using it in a large animal or human, you turn up the current. I refuse to make a tinier one that would only be good for rat studies. So here's just some pictures of the core shell nanofibrous device. It was super convenient. They were already using an annular device with a hollow core, because we attach the oxygen generator to the hollow core and use that for the delivery. And there's a quick picture of the device bending and flexing. So to some of the results that are shown in the paper, we started the work with in vitro testing using human islets, rat islets, and we also used INS, we also used cell clusters. So the basic setup is you take a cell capsule, you put it in a 1% oxygen incubator, so that's roughly mimicking the subcutaneous space. So the ambient environment is low oxygen, and then you either turn on or don't turn on the oxygen generator. And we also, under normoxia, we put capsules into a regular incubator, 21% oxygen. So basically, we were seeing that in a 24-hour in vitro bench study, that if you did not have oxygen, you turn the oxygen off, and they were in the low oxygen incubator, you had damage to the cells, and no insulin secretion, and very, very little insulin staining. So here you see low insulin staining in the hypoxia. You see low insulin staining and good insulin staining in both the normoxia, that's a positive control, and the hypoxia condition with added oxygenation. So we've done insulin secretion studies, we've done IPGTT studies in this, and we've gotten good transport properties. And besides the animals, I'm getting to the animals in a moment, sorry. In the in vivo rat studies, we did this work in a rat. I will admit to you that wearable electronic packaging is challenging. We definitely had animals chew through the cable, and then they went back to the control condition. We are doing this in immunocompetent rats, so these capsules are truly allogeneically protective. So some of the initial work without oxygenation is shown on the left. The animals were made diabetic by STZ, and you see that the blood glucose stayed high through the whole time. In the middle, you see some of our difficulties. So basically, rat D1, D2, D3 were implanted, they were given oxygen, and the oxygen stopped because of a problem with the wiring or the tubing. The rat got out of its jacket, etc. And then their blood glucose dropped for the first day or two, and when they had the problem, blood glucose went back up. And on the right, we have the cohort that's presented in the paper. Although I will say the MAW lab is very, very open and transparent. They will show you all their data. So they're showing the ones that worked and the ones that didn't work with explanations. We had many devices shown here. All the 6 rats that had continuous oxygenation had diabetes corrected within a day or two of implant, and all the failures were due to something logistical. So there was no animal that reverted to high blood glucose that we didn't have a reason that had happened. And then the ones that had oxygen stopped, we did explants on those and looked at the histology. And then, as I said, at 5 weeks, any animals that were running at 5 weeks got an IPGTT glucose challenge test. And the good news is that the glucose response was quick and fast. There's been discussions in the field. What if you control average blood glucose, but minute-to-minute blood glucose is all over the place and you have bad outcomes because your A1C levels are through the roof. So we're not seeing that. I'm sure there's some delay of transport through the membranes, but it hasn't caused any remarkable problems. Just a little bit of microscopy shown. So on the bottom right, at day 45, you see nice rounded pancreatic islets. So again, we've done some staining. You'll have to look more carefully at this in the paper. So our next generation is going to be a fully implantable system, and the system after that will be clinically ready. So we think we can take the components of this system and make an implant about the size of a credit card. We have already been working on eliminating the hydrogen, so the hydrogen can diffuse safely into the tissue through a diffuser design, and we've done a little bit of first preliminary studies for that in a rat and in a pig. We have a board design ready with a manufacturing partner, and we're looking for grant funding and venture funding to implement some of these things. We think we'll be able to get away with once a week, weekly recharging. So the only thing the patient would have to do is put a device, a band, or sit in a chair that has a wireless recharger. So it's like charging your phone. There's nothing coming through the person's skin. Their skin is totally intact. As you know, there are other devices like this. There's neurostimulators that charge through the skin. There's one glucose sensor that reads when you put a power pack against the skin, etc. So again, we're working on proof of concepts in the porcine model. Everyone knows the porcine model is much better for the subcutaneous space, more realistic compared to the loose-skinned rats. And we'd like to do stem cell-derived islets with partners. So instead of only using cadaveric islets, we'd like to move to stem cell partners. We will be working on a fully implantable system with no external electronics for the pig model. Just the acknowledgements, a large group of people at Giener have worked on this. I'm showing the people first that are working on it now, later the people that worked on this historically and generated some of the data. Both Dr. Pham and Laura Tran are the authors that did the majority of the animal work. And right now, Mr. Okada and BJ Kim are working on this project now in Bo Wang's lab at Cornell. And our advisors include Simon Stone on electrolyzers. Dr. Shapiro is following this work with us, and he's published with Mingling before. And Doug Lawrence is a former Becton Dickinson executive. And we've had good funding through Breakthrough, NIDDK, some pharma partnerships, internal monies, and so on.
Thank you very much, Linda. That was a great presentation. Very optimistic in its projections, I'd say, and which is good news. I did want to ask you a little bit about some of the, kind of like lead up learnings from other companies. So for instance, Vertex, has had a bit of a retreat from their islet device combo. That kind of creates a strategic opening for you guys. But, as you sort of mentioned, it appears that maybe the VX-26-4 trial might had poor long-term beta survival because it might have been missing like the oxygen link. And if we think about other companies that were in the space, Beta O2 or Beta Air, that was developed in Israel with MIT collaborators, they had an implantable alginate slab of islets plus an oxygen chamber. So they were trying to rectify that. But their results in the clinical trial resulted only in minute circulating C peptide. So they had good cell survival, but poor functional performance. I guess I would ask you to kind of just touch on, what kind of opportunity this gives Persista to kind of fill in some of the blanks.
So I think there's a number of companies that are working heavily on the stem cells, and a number of them are trying hydrogel only solutions. So they're just trying to encapsulate the cells in hydrogels. And for Th1nk in Canada, their trials have not been working, even though they're partnered with Evotech and in the future could be getting stem cells from them. I'm not too optimistic about the hydrogel type approaches. I think if we want to get something in the clinic quickly, I think we shouldn't mess around with immunoprotective hydrogels and hydrogels that may be unstable mechanically and physically. And I think we should just put a membrane there. So that's where we're going. So my goal, Monica, is to keep increasing the proof of concepts with this device, increase our readiness to produce larger quantities of the components and eventually a fully implantable system, and keep waiting to partner with people. I'm also talking to some of the groups, not those major folks, but other groups that have pancreatic islet stem cell lines, either available for license or commercially available, to partner with them. So they're not planning to take a product to market. They're supplying these to researchers, et cetera, like the Army Lab. There's a bunch of other groups. Obviously,
right, they're in the basics. I mean, I guess one of the biggest issues is in any of these implants is people are trying to mitigate the fibrosis component. I mean, I think you've spoken to that a little bit more. I didn't know if you could extend that.
Yes, so the Cornell approach and the patents we've licensed from them, there's several layers of protection that we could use against fibrosis. So our goal, the material seems to be fairly anti-fibrotic. You can put a hydrogel on it. There's hydrogels we've licensed from them that are more anti-fibrotic. So we'll only put as many layers or use as many tricks as we have to. So we are looking forward, Monica, to seeing some porcine data just to see how these act in the porcine model. And I know that there's a lot of controversy about different porcine models are more and less fibrotic. So that's basically to be found out. Shaheen, you had a question earlier?
Could you go to that histology again?
Yeah, Shaheen, I'm not the one to delve into the histology with you,
where are the blood vessels here? How does this insulin glucose get detected, and where are the blood vessels that are in close opposition to this?
Shaheen, in this particular experiment, this is the capsule explanted only. So it doesn't have the surrounding tissue around it. Right now, the Cornell core is trying to do cross sections from block excisions that have tissue and the gels. So blood vessels are not going to get inside the annular area where the cells are because we're blocking blood vessels. So we're counting on diffusion in and out of the capsule and close blood vessels, and we're counting on extra supplemental oxygen to give more oxygen. Remember, oxygen is available in the body in micromolar quantities. Most of the other nutrients are in millimolar quantities. So I don't know how we're an oxygen breathing species, right? Because oxygen system and hemoglobin, oxygen is very short in supply. So Shaheen, that's upcoming work where we're going to do cross sections through the whole device, including the tissue, and look for capillaries near the device. So, many people have tried, and Th1nk is trying to get lots of blood vessels in the tissue and then stick the device in, right? Thinking that might be enough. We don't think that's enough. We think the added oxygen is the rate limiting nutrient. But if you have any protocols, Shaheen, that you've seen people use for counting the blood vessels, et cetera, I'm using an old Baxter protocol of how you count how many vessels there are, how close to the device. But if the Cornell lab can't do these cross sections, I've had a private commercial lab in Lexington, MA, CB set, they've done plastic block for me when I was doing Therasite type devices. And Therasite devices are hard to cross section too, right? Because of the tough PTFE of the Teflon type material. So we are working on showing the blood vessels, but in this histology, it's all the sex cell capsule explanted, opened up and looked at. So good point.
The reason I'm asking you that is because I think I mentioned that during the meeting. It's not just blood supply. Lymphatic system has to be there. What happens over time, cellular debris, cellular processes release a lot of waste material that are not diffusible. They have to go through lymphatic system. They
Yep.
have to negotiate through. My worry is that over time there is a process in vascular biology called saponification. All these abnormal membrane proteins are going to, you see it right now around you see those white stuff? Those are membrane fragments, saponification, that when the MIT group with Doug Melton did that, they put it aside because they found out no matter what membrane they used, besides what Monica said about fibrosis, thing didn't work out because there is no cell in body that is not directly vascularized. We just published that. It's not just that the blood vessels deliver oxygen, they provide angiogram factor, growth factor for the cell. There is a paper that's going to come in Dev Cell shows that the BMP2 from the blood vessels, from stroma, is the key to survival. So I just worry that over time, if you don't have blood supply to avoid immune, I totally understand that, but the debris and cellular product is going to suffocate this if you cannot take it out. And the problem is that the more oxygen you have, blood vessels are not going to come in. HIF goes down, VEGF goes down, angiopoietins goes down. We all became this big organism because hypoxia induced vascularization.
For
So
sure.
it's amazing that it's working. I want to congratulate you, but I am at awe as a vascular how this works. I just don't get it.
Yeah, no,
Because
and there's some thoughts that the beta O2, the one or two patients in those trials, there was some debris in those that was thought. I mean, I think they may have even had amyloid formation.
yes, yes, amyloid. You see, this is a lot of amyloid. One of the biggest, exactly. That's what I was going to say, amyloid and all of that stuff. But that's why, most of the data come from Alzheimer's shows when the blood vessels are damaged in the brain, in subset of Alzheimer's, that's how you build up amyloid. And that causes suffocation and all of the debris. Blood vessels are designed to remove amyloid. Lymphatic system, all of those work together to do that. So if you can bring some blood vessels in, I mean, this is your product, your patent, but
Well, that's what that's what Viasite did, right? They poked holes in their membranes and let blood vessels go in their second trial.
no, I know it's not enough.
Yeah,
You,
yeah, okay.
you,
So
okay,
she, so
let's
he,
say,
what are we left with? Waiting for immunoprotective cells, right? So I'm trying to do a first generation, and I hope we can get cells that are okay to allow tissue integration and not use immunosuppression.
no, I totally understand, but 90% factor produced by stroma, pericyte, and endothelium, like DLL4, thrombospondin, they are membrane bound. None of these get diffuse. Every stem cell and cell in our body is direct cellular contact to get those signals for survival. There are many papers shows that the notch signaling, BMP, all of those requires direct cellular contact. I am very curious to see in your system whether these endothelial cells have survived in those. That would be amazing if you do the staining with Ming Ma, to see whether there is some endothelial cell, because it's amazing how they're surviving.
Yeah,
Where
yeah. So
what
we'll work
factors are
we'll
coming
work
from?
Shaheen on getting cross sections so we see the tissue and the capsule in
Yeah,
situ. That'll be important. Can
yeah.
I take questions from anyone else? Shaheen and I can have conversations offline all the time.
No worries. Yeah, we've had a number of people on here. Antonio Citro, where they're going after the lung type model. We've had Emir Dolan from Galway talking about implantable systems. And we've had representatives we've had Mingling Ma, we've had representatives from Vertex, and ViaCyte. And so it is really important, I think, to keep the conversation going, learning from the clinical lessons and history of those that have come before, and sort of keeping the science, solving for usability and durability. I mean, it's really, really impressive that people are continuing to press forward strongly into this very difficult problem set. So Linda, I really appreciate your talking today, and fingers crossed that this can proceed in all the right ways. I really appreciate it.
Yeah, I just put my email address in the chat. People can ask questions. And
Fantastic.
of course, there's a lot more detail in the paper, and some of my
Yeah.
collaborators can explain how the histology was done better than I can.
Well, we'll look for the next histology paper coming out once you do those experiments. And thanks again for joining us. Really appreciate it. Have a great rest of your day.
Yeah, thanks everybody. Appreciate it.